You might have noticed that whenever you get injured while playing, the wound gets healed after a few days. How? When we are injured, the cells of that place get damaged. Soon after, the body starts the healing process in which the intact cells present around the wound start dividing and fill the wound with newly formed cells. Similarly, when we pluck flowers from the plants, they also get injured and the wounds are healed by the cell division in the surrounding cells. Cell division is one of the very important characteristics of cells and living organisms. Multicellular organisms grow and increase in size due to cell division, which increase the number of cells in them. Emaciated body can be restored by cell division. New individuals of a species are formed from existing ones of the same species by the process of reproduction, in which cell division plays a vital role. Cell division is of two types: mitosis and meiosis. Mitosis occurs in somatic cells and stem cells of the body, whereas meiosis occurs in germ cells only. We have studied earlier about the structural organization of a cell. We know that each cell has a nucleus and many cell organelles present in the cytoplasm. Before any type of cell division, the cell prepares itself and in this process doubles up its chromosome number present in its nucleus. That is, if the number of chromosomes is 2n, then it is doubled up to 4n. We have studied about chromosomes in the previous standard. Chromosomes are made up of DNA and appear dumbbell-shaped midway during cell division. Do you remember the name of its various parts? Come, let us label each part of chromosome. We all know that each chromosome is made up of DNA. The constricted region on each chromosome is called primary constriction or centromere, and each part of chromosome is called arm, P-arm and Q-arm. Come, let us now find out how exactly chromosomes take part in cell division. Mitosis. Somatic cells and stem cells divide by mitosis. The mother cell which undergoes cell division is 2n, that is, diploid, which means the chromosomes present in the nucleus are in pairs. Before cell division, chromosomes are duplicated and their numbers are doubled up to 4n. The whole process of mitosis is completed through two main steps: karyokinesis, that is nuclear division, and cytokinesis, that is cytoplasmic division. The division of nucleus, that is karyokinesis, is completed through four steps: prophase, metaphase, anaphase, and telophase. Let us learn about them one by one. Prophase. In prophase, condensation of thin thread-like chromosomes starts. Due to this, they become short and thick, and they start to appear along with their pairs of sister chromatids. Centrioles duplicate, and each centriole moves towards the opposite poles of the cell. Simultaneously, nuclear membrane and nucleolus start to disappear. Metaphase. Nuclear membrane completely disappears before the beginning of metaphase. Chromosomes complete their condensation and become clearly visible along with sister chromatids. In metaphase, all chromosomes start arranging themselves parallel to equatorial plane, that is, central plane of the cell. Special type of flexible protein fibers, also called spindle fibers, are formed between centromere of each chromosome and both centrioles. Anaphase. In anaphase, centromeres split and thereby sister chromatids of each chromosome separate and are pulled apart in opposite directions with the help of spindle fibers. These separated sister chromatids are called daughter chromosomes. When each set of chromosomes reach at two opposite poles of the cell, they appear like a bunch of bananas. Telophase. The chromosomes which have reached the opposite poles of the cell now start to decondense. Due to this, each of the chromosomes again become like a fine, delicate thread and start disappearing. Very soon they are invisible. Nuclear membrane reappears around each set of chromosomes at both poles. Finally, two daughter nuclei are formed in a cell. Nucleolus also reappears in each daughter nuclei. Spindle fibers completely disappear. By the end of telophase, karyokinesis is complete, resulting in the formation of two nucleus with same number of chromosomes. Now the second step of mitosis, that is, cytokinesis, begins. Cytokinesis. In cytokinesis, a notch is formed at the equatorial plane of the cell, which deepens gradually towards the center, and finally two new cells are formed. These newly formed cells are called daughter cells. However, in case of plant cells, instead of a notch, a cell plate is formed exactly at the midline of the cell, which grows towards the periphery, and thus cytokinesis is completed. After a detailed study about mitosis, can you tell why this process is considered to be so vital? Mitosis is essential for the growth of the body. Besides, it is also necessary for the restoration of emaciated body, wound healing, formation of blood cells, etc. Come, let us now proceed to learn about another type of cell division, that is, meiosis. Meiosis. Meiosis occurs only in germ cells. The process of gamete production and spore formation occurs by meiosis. After meiosis, one diploid cell gives 4 haploid cells. Meiosis is completed through 2 stages: meiosis 1, meiosis 2. Meiosis 1. This process completes in several steps. During this cell division, crossing over occurs between the homologous chromosomes present in the 2N, that is diploid mother cell. Homologous chromosomes are chromosomal pairs that are similar in length, gene pair, and centromere location. Crossing over results in genetic recombination. Thereafter, these chromosomes divide into 2 groups, and finally, 2 haploid daughter cells are formed, each with n number of chromosomes, which means each cell possesses only 1 copy of a chromosomal pair. Meiosis 2. Meiosis 2 is just like mitosis. In this stage, the 2 haploid daughter cells formed in meiosis 1 undergo division, and finally 4 haploid daughter cells are formed. Since there was genetic recombination in meiosis 1, all the 4 daughter cells formed are genetically different from mother cell and from each other. These cells now transform into gametes which are always haploid, that is, have n number of chromosomes. During fertilization, male haploid gamete joins with female haploid gamete, and a diploid zygote is formed, which divides and grows into a new diploid living organism. Therefore, formation of haploid gametes help the living organism to maintain the number of chromosomes in its species. After learning about cell division, let us do an interesting activity to observe different steps of mitosis in onion plant. Activity: take a medium-sized onion and keep it in a glass jar filled with water in such a way that the roots of onion remain dipped in the water. New roots develop within 4 to 5 days. Cut the tips of these roots and put them in a Petri dish.
Add few drops of iodine solution.
Take one root tip on a glass slide and press it with the help of forceps. Add 1 to 2 drops of water on it and cover it with a cover slip. Observe the prepared slide under the compound microscope. Which step of mitosis do you see? We see metaphase step in the cell of the root tip. How did you identify the step? As all the chromosomes are arranged parallel to the equatorial plane of the cell, we can say that the cell observed is in metaphase step. Sketch the figure in your notebook.